No, I can't. It's gone.
None of it came through. Oh, it did. This much of it did. And it stopped.
Wednesday, March 27, 2024. I'm Vincent Racaniello, coming to you from the Incubator in New York City. I never say that, but I thought. Well, I started a new stream.
How do we get them all on it?
They all got a notification.
Okay.
Sorry about that. For
Jesus.
some reason, the stream stopped. And so I just started a new one, and it's hopefully...
Do the intro again.
All right, let's do the intro again. We lost all the intros and hellos. I'll start all over again. Office hours. Here we go. From Microbe TV, this is Office Hours for Wednesday, March 27th, 2024. I'm Vincent Racaniello, and welcome to your viral corner of the internet. Joining me tonight here in New York City at the Incubator is Karen, our studio regulator. Hello, Karen.
Hello.
Maybe you were responsible for the stream
And
stuff.
maybe you can always blame me. I'll take it.
I hope everyone has found the new stream. If you are, some button get pushed. We have to figure out how Karen can sit next to me and participate in this without having a problem. So I don't see any moderators, but I know there were moderators here before. Let me see if I can text them. Let's see. I shouldn't be doing this. I shouldn't be texting during a live stream.
You shouldn't be texting.
Well, there are no mods on this. And the moderators who were here before, maybe
They'll
you
show
can,
back.
maybe you can
I'm looking at stuff.
tell them. We had Steph. We had Andrew from New Zealand. I saw Tom. I saw Frank. I believe that those were the moderators who were here tonight. And let's see where everyone was from. So basically, the original stream stopped. I don't know why. So I just started a new one. And I didn't have time to put any information in.
You can fix it in post.
We'll fix it in post. So I restarted, but none of the mods are here. Oh, Andrew is here. Okay, Andrew, thank you. At least we have one moderator, although I don't think it's so essential. But tell us where you're from, folks. Let's see. Ooh, B-E. It's from Algeria. How do you say that? B or B? What do you think from Algeria? B-E.
B-E?
B-E? How would that be?
Bah?
Be. Tell us if you hear Karen, by the way, just to be sure.
Bah?
You hear Karen sound bah.
My question becomes, when we did the TWEVO, how did you log on to the same eCam live?
It's the same eCam.
And you opened eCam and then did what?
I was just watching it because I had.
It just showed
No,
up?
actually, I wasn't on eCam. I was watching the stream like you were. You were on eCam, and I would tell you to select certain questions. You were running the eCam stream, so we can't
there
have
was
two.
still a delay.
Yeah.
Okay, that's what I thought. You were on eCam because
Yeah.
you were seeing all the. Okay.
Anyway, welcome from Algeria. I don't know if anyone's ever been here from Algeria. That's fantastic. Kathleen, great intro. Well, that intro's gone. Thank you. From South Jersey, yeah, when it's raining. Let's see. Who else is here? Noir is from Santa Fe. Abdul Aziz is from Saudi Arabia. I know that. Ioto 2001 is from Cincinnati. Maureen is from Northeast Ohio. Andrew is one of our mods who is in New Zealand. I guess the other mods left. Carol is from Southern California, our favorite nephrologist. You know, guys all know what a nephrologist is, right? Someone who takes care of your kidneys.
I have a personal relationship with some nephrologists.
I'm sorry. On the other stream, they said you started another stream. I made the switch. Good. I'm glad they did that. Joseph is from Ontario, Canada. Doreen is from drizzly Hartford, Connecticut. Meg 378 is from Chicago, Illinois. So the way you say it is Betty. Oh, Betty from Algeria. Welcome. Jonathan from Encinitas. Richard from Portland, Oregon, where my puppy is barking loudly. Noir is from Jeremy, New Mexico. Bernard, where are you this week? Claire is in the UK. Pete is in London. Karen is audible. Thank you very much. Pete, Bernard said you're audible.
Oh, that is more than I can ask for.
And Doreen says you have a great broadcast voice.
Oh, thank you.
You can call me Batul. Okay, Batul. Welcome from Algeria. It's fantastic that you're joining us tonight. John is from Minneapolis. Hat is from Los Angeles. Carl is from Waterloo, Wisconsin. Tom is from Salt Lake City. Nicola is from Italy. Older and Wiser is from Cleveland. James is from Longmont, Colorado. F. Yang is from Houston. I didn't select him. There we go. Zan is from Dunbogan, Australia. MK is from Eastern Massachusetts. EHS is from Eastern Pennsylvania. Tom, welcome back. Thanks for coming from the other stream. You're in Oregon tonight. Greenfields is from Ireland. I'll get to your question as soon as I see where everyone is from. Troy is from British Columbia. Bernard is in D.C. this week. Cool. Oh, Otto, my dad worked for Sabin in college in the 1950s. That reminds me, Karen, remind me to make a plea for Salk information. Can you write that
for
down?
For what? How do I spell it?
Salk, S-A-L-K, information.
Salk, info. You got it.
Calliope. Where's Calliope? Is somewhere in Canada, if I remember. Toronto, maybe.
Hey, Calliope. Look, she said hello to me, too.
She said hello to you. Vincent and Karen, that's right. And Bus Driving 1 is from Grand Rapids, Michigan. I don't think we're going to have as many people because probably people are going to the other one. But this is live on the YouTube page now, so they should come here.
But and also, even if we don't have as many people, it just means it's more intimate.
That's fine. You know, I'm a numbers guy.
I know.
Wayne is from Urbana, Illinois. Yeah, Will is from Changsha, China. You found it. Yeah, there's a 29 seconds. I should probably delete that. I will delete that 29-second thing later. I don't need to do it right now.
I might be able to do that right now.
Yeah, you are, since you're an administrator.
I can do all kinds.
But don't do the wrong one.
I know, right? That would be bad. Twice in a row.
Vanity, welcome back. Vanity is another of our moderators. I forgot that you were in the other room, so thank you very much for being here. Elizabeth is West Virginia. Jessica is in Toledo, Ohio. Nova is in Montreal on vacation. John is in Torrey, Utah. Good memory. Yes, you are in Toronto. Sometimes my memory serves me. Other times it fails me. And at the most inopportune times. Eliza is from Toronto. Is this my talented voice person? Yes. That's Karen. Karen is the studio, is the incubator regulator and our road manager, right? So when here at the studio, she manages things that happen. And then when we go on the road, she manages that. She carries all the equipment, sets it up, records, makes sure the audience is enthusiastic, and then breaks it down and brings it back home. All good stuff. Thank you, Karen.
Thank you.
Nebby is from Florida. David is from Barbados. Wow. That's pretty cool. Must be nice and warm down there. All right, that seems to do it for now. I mean, there'll be more people coming, and we'll try and highlight them. But I wanted to do a little mini lecture at the outset here, and then we'll take your questions. Okay, folks, does that sound like a good plan?
Okay, I'm deleting it now.
Lucy is from the Sunshine Coast of BC. Welcome to everyone. We're really happy to have you. And yeah, go ahead and hit the like button. We have 112 people and 38 likes, so please hit the like button. Elizabeth likes the name Incubator Regulator, so Elizabeth understands that.
Elizabeth, I went out with Vincent when we were on the road at one point, and there were students with us, right? And I really don't want to fall into some weird capitalism moment here. So I asked them, I don't want like office manager, studio manager, like we should be a team. And so I asked them, what is essential in an incubator that has nothing to do with having a degree in science? And they said, what, Vincent?
Regulator.
They said, I think it was CO2 regulator.
CO2 regulator,
technically,
yeah.
but we felt like that got a little tedious, right?
Yeah, we don't need CO2 in there. People will
Nah.
not really know what's
Understand
going
that
on.
it's a little much.
All right, so one more person here just popped in, for James from Austin, Texas. Welcome. All right, now there is a way for me to show the slides and have my face. Do I dare to try it in real time, Karen?
I think we should dare everything now because we've already screwed up worse than is humanly possible.
This is the worst start of office hours ever, but it's...
Which means we have no place to go but up.
We
Right?
fixed it. We're on, and
fixed it.
people came,
Right.
120 people, and we're here. So there's a way to put a camera overlay here. Here it is, a camera overlay. I got it here, and all I have to do is bring it. I don't think you can have a camera overlay with a slide overlay. That's the problem. Yeah, you can't have that. So you can't see me. Actually, I'm sure you can, but I don't know how to do it. Where's the overlay again? Yeah, here it is, the camera. You know, the problem is there's a delay. Oh, I know there's a way to layer them,
Oh,
right?
that looks cool.
It just made a noise. Sorry for the making the sausage, folks. We are trying to figure out how to have the slides and my face in it because I get very animated when I talk, and
Right.
you should see it. So here's like an overlay. You probably see double me now. Oh, yeah, it's just like a delay. That's the problem. It's
Right,
gonna be a
right.
delay.
I saw you in a little box for a minute.
You did?
Mhm,
Okay, so
I
that's
did.
the overlay.
And then
But-
I think
But you didn't see the slides, right?
not yet.
So here's screen share. And all you're seeing is my stinking screen there. That's not what I want. I want you to be able to see the slides.
Okay, now with the delay, I got your face up again.
Yeah, there's a face inside of a face, but that's not what I want. I want you to be able to see this screen.
I'm going to come look at your laptop.
You see, this is just the stream that I have here, but this is the screen, and this I probably have too many
So
monitors. That's the
when
problem.
we did the TWEVO, we pulled it up, we added it in
Well, I'm
eCam.
not streaming from here. It says this.
And-
I'm streaming from here.
So we added it in eCam
Yeah.
as a file, right?
Added what in eCam as a file?
Well, all the extra screens that we added.
You see, now all we see is this. So can we get... You see, there's a desktop under it. There's just no way that we're going to see me. I don't know why it's doing that. Screen share. So let's turn off this. Well, that does... See, that just turned off the
Camera.
camera. Let me turn off the screen share again. And you see, there's the little camera. See?
Oh, so when you screen share, you get no cameras.
So I want to bring this over here, but this thing is in the way. So if I take off the screen share, right? Let's have to hide this. The screen share is coming from here, I think. What is this thing? Oh my gosh. All these things, I have no idea what they are. Sorry, folks.
Well, how about we just you show them the screen you want to share, and I'll describe your animation in audio background.
I don't think that's going to work, but let me try screen sharing again. Is there a picture-in-picture function? Yeah, that's what I'm trying to get, but the problem is that when I have the lecture slides up, like I do now, there's no way to overlay a camera on top of it. The camera goes behind it, and I mean, I could try moving it below. Oh, look!
What'd you do?
I got it to work. I
Excellent.
dragged it below the screen share in the list.
Excellent.
So it's a that's how it works. Can you see me now? The little me in the
Not quite yet.
it's going to come up in a few seconds.
It will.
And then you'll see me and all my glorious animation. I think I can make it a little smaller here. Yeah, there it is. Holy cow, it works.
Oh.
How does it look? Is it big enough for everyone to see? That's the question. Because last time,
So right now, what it looks like, I'm going to show you my screen, right?
yeah.
Right now, I've got some background that I don't need. Can you blow that up? Can you blow up the thing that you made?
Oh, the slide?
Yep. Can you make the
Yeah,
slide bigger?
I can. Let's see. Yeah, I think you have to hit.
We don't have to master this right now. We just have to get it to a point to where it works for you.
How's that? Is that better?
Probably.
Now my picture is too big. Let's try moving it up here.
There you go.
Right now you can see me.
Yes. It's beautiful.
All right, how's that, folks? Are we good? Now I want to know where do you see the pointer? Yeah, that's getting there, and it's moving up. Right. So I think the arrow is going to be the thing. Yeah, there it is. Okay, folks, we're ready.
I was going to say, I'm pretty sure nobody here is asinterested in learning about this tech as much as we are. So maybe at some point we just.
Well, people say
up.
it looks good.
Okay. All right. That's
Right?
all that
And
matters.
They're happy that we
I think you
figured
should.
it out.
I think you should switch to the blue glasses first.
Okay. Are you serious or are you just joking? Are you serious?
I'm just going to ask you to switch glasses
All right.
every 20 minutes.
So what I want to talk about today are herpes viruses, just herpes simplex viruses, because we really have a good understanding of how they establish latency and they come out of latency. So first, what am I talking about? So with virus-host interactions, we talk about two general patterns of interactions. There's the acute infections and the persistent infections. So today we're going to focus on persistent, but I just want you to know the difference. Here's a slide which contrasts these two patterns. We have acute on top. And on this graph, the y-axis is virus production. That's the axis on the left. And the x-axis, which is the axis on the bottom, is time. Okay? And the blue line is virus production, and the red bars are periods of disease caused by virus infection. And on the top, you see a typical acute infection where you have rapid onset of virus reproduction after introduction into the host. You have symptoms develop, you shed virus, and then it's resolved. Either you clear the infection, your immune response takes care of it, or you die, unfortunately. So either way, you do not have the infection your whole life. It is a certain period of time. That's why we call it an acute infection, because it's on and off. And in contrast, the persistent infections, they are with you for your whole life, right? You get them at a certain age, typically a young age, and they're with you forever. They may cause periods of disease or not. And here at the bottom here, there are several different kinds of persistent infections. The second line here is the herpes virus, which we call a latent infection, because there are periods when there's no virus produced, and then there are periods when there is virus produced. And sometimes there's virus without symptoms, right? Whereas there are many viruses that infect us always and reproduce all the time, and we shed them, and we can talk about those another time.
Is chickenpox also that?
So chickenpox is a herpes virus, so it would be more like
it is a herpes virus,
but more like herpes simplex, yeah. But this third bar here, the persistent asymptomatic, JC virus, anelloviruses, many viruses infect all of us. We shed them constantly. Our blood is full of them. They cause no disease, and we don't really understand that. But we're not going to talk about those today. And then finally is the persistent pathogenic long viruses like HIV, where you have an initial acute event, then you have low levels of virus for many, many years, culminating in the appearance of AIDS, and if you're not treated, death. So that's a very different kind of infection. But today I want to focus on the latent infections, the herpes viruses, because we really understand a lot about that. Now, herpes virus, let me get this so it fits in the window. What's going on? Here we go. Now, apparently I have to drag this over a little. Nope, no good.
Can you make the screen full screen?
No, you can't. If you do that, everything disappears. We'll have to test this another time, Karen, okay? We need to do some
I'm putting it on the list.
testing, some serious testing. Now that's kind of small. Yeah, I can't make it full screen. That would be the ideal way to show a presentation. Anyway, let's just go here. Here's herpes virus, some of the points I want to make. The viral gene products that promote replication are not made or they're found in low concentration. So viruses need to make proteins so they can make more of themselves. And in a latent infection, they're often not made. Whereas in a persistent, asymptomatic or pathogenic infection, they're always made all the time. Yeah, I know. If you put it in presentation mode, it's not going to work. Watch. Now I'm gone, and there's nothing. I'm going to wait a few seconds for the delay. And you're going to see it's not there. So the full screen doesn't work with this eCam. All right, I'll close the style guide on the right. Fine. Yeah, that's probably right. Thank you very much. Yeah, that's the key. Close this. Now we got all the words here, I think. Well, not quite, but
that's
it's
good.
still running off to the left here because it's too big. I have it zoomed in 75%, but I can't. You see I'll move this over here.
It looks good, Vincent. It's enough.
Yep. Well, I'm getting a lot of suggestions.
Yeah, but eventually.
But Nova Guy's suggestion to
close the style guide.
Yeah,
that
was
was
very
pretty
good.
Smart.
All right. So the other reason for having a latent infection is that cells that have the latent genome, they're poorly recognized by the immune system, and so the viral genome persists and periodically undergoes productive infection, so that you can get virus spread to new hosts. Because if the genome just went into a host and sat there and did nothing, it would be the end. When that person died, the virus would be done, right? And that would not be a successful infection. So herpes simplex viruses, there are two, herpes simplex 1 and 2. These, over 80% of Americans are seropositive with viral DNA in your peripheral nervous system. I'll tell you what that is in a moment, but it's outside of your brain and spinal cord. Outside of that, that's the central nervous system. Your peripheral nervous system is everything else outside of it. So over 80% of us have been infected with herpes, and you have it for the rest of your life. You can't get rid of it. Like I say all the time, unlike love, herpes is forever. Isn't that nice?
And sometimes it comes with love, right?
Yeah, of course. It does come. But usually it's your parents kissing you when you're very young and they have it in their saliva, and that's it. You get the infection. So millions of people, not just in the U.S., of course, but everywhere, carry latent viral genomes in their nervous system without any symptoms. 40 million people experience recurrent herpes disease, which are cold sores of various kinds. And when you undergo stress, as we'll see in a moment.
I'm going to make a T-shirt that says, "My mother gave me herpes."
Yes, she did. Mm-hmm. She did. And then there are two, herpes 1 and 2. Herpes 1 is generally the oral pathogen. You get it from kissing. And herpes 2 is the genital pathogen, which you would get from sex. But as you know, those two areas can mix sometimes.
You can get 2 on your mouth. You can get 1 on your genitals.
You can get 2 on your mouth and 1 on your
Sure.
genitals. That's right.
If you work really hard at it.
You don't have to work hard at
don't?
all, actually. It's pretty easy. You just have to find a willing partner. Okay, so we have 2 different herpes viruses, but they're all going to do pretty much the same thing, just in different anatomical locations. And I think this is a characteristic of a well-adapted pathogen. We have the virus is able to remain in you without causing disease most of the time. Periodically, it causes a bit of disease, but in some people, it doesn't cause any disease, and it spreads to new people. So that is beautifully adapted pathogen. So how does this work? Here's your first herpes infection. You're often infected in utero or during birth because the mother has herpes viruses there, or after birth in the first year of life. You know, your parents are shedding it in their saliva. They kiss you. It's now on you, and you get it. That first infection, incubation period, It's usually an inapparent infection, which means there are no symptoms. But sometimes you can have fever, you can have sore throat, you can have ulcerative and vesicular lesions in your mouth, you can have all kinds of things. And that infection lasts maybe a week or two. And what's happening during that infection? So you've got virus coming into your mouth. Here are virus particles, and they're going to infect the epidermal cells, the cells of inside of your mouth, let's say. This is epidermis and dermis. So this is skin, which could also happen, but it could also be inside of your mouth. The viruses infect those cells, they reproduce in them, and then they exit the bottom of the cell. And just below that surface are nerves, sensory nerves, right? And so the virus enters the end of the sensory nerve, and the genome goes all the way up to the cell body of that neuron. So this cell is one long neuron, right, with axon and dendrites, the two long processes. And then there's a cell body. And these cell bodies are typically from many neurons in one place, and we call that a ganglia. I have a picture of that in a moment. You'll see. So the virus then becomes silent in that nerve cell. No more virus particles are produced until reactivation, when more virus is produced and the virus leaves. So that's the overall picture. And we'll go into some of the details now because it's really interesting. Doreen says, maybe we all need blue glasses for Wednesday office hours.
I think that
we should. Everybody has to wear blue
We're
glasses.
now going to market Microbe TV blue glasses.
All right, so here's a schematic of the epithelial layer, right? 3 cells. The virus has reproduced through them. It enters the nerve ending, which is shown here, and travels to the ganglia. So here is one neuron with the nucleus, and you can see the herpes DNA there, that little squiggle. And there are other nuclei here. There are other neurons in this ganglia. So ganglia have many neurons that are connected to nerves that go to various places, right? So the genome enters the nucleus, and it's silenced. The nucleus says, silence herpes genome. No more gene expression. No more mRNA made, so no more proteins are going to be made. And there are many copies of the viral DNA. And we call it episomal, which means it's separate from the chromosome. Episomal, I don't know if that's a real word in life, episomal. I bet it's not.
Does it have anything to do with episodes?
No, it has nothing to do with episomal. It's a specific word for this DNA. There's no English word of episome. Episome means DNA, a piece of DNA in the nucleus separate from the chromosome. So that's why it's shown as that little squigger. And now that DNA does not have to replicate because neurons do not divide, right? If cells were dividing and the virus was going to go to each cell, it would have to reproduce. But here the neurons don't divide, so they're okay to be quiet. And this is forever. This is the forever part. You can't cure this latent infection. There's no way. You can take all the antivirals you want. It's not going to affect the piece of DNA sitting in the nucleus. So that's true. Unlike love, herpes is forever. Now, if you go to my YouTube page, right, and you look up my herpes lectures like this one, you will see lots of comments below of people saying, Dr. So-and-so cured me of my herpes. It's not true. No doctor can cure you of your herpes. It's don't spend money on it because it will not happen.
Well, now you can also, can you not have herpes and never have symptoms?
Yeah, some people never. I've never had a cold sore in my life.
Me neither.
I'm sure I have herpes in my
I know I
peripheral.
do because I applied for, back in the olden days, a project where they were using people to study them for an actual genital herpes, a simplex 2 vaccine, but you couldn't have simplex 1 in order to be in the study. So they gave you a test,
so
and that's how I know I have one, but I've never had a cold sore.
So this is good. Wayne says epi means around or near, and some means body. So there you go, around the body. Thank you very much.
It's Latin, yeah.
Yes, so everything comes from the Latin and sometimes the Greek as well. All right, so our herpes this is the latent stage, right? Latency, the genome is sitting there in the nucleus. So what's happening next? Well, first of all, I told you there's no gene expression, but there is an mRNA that's made. There is an RNA that's made. It's called LAT, latency-associated transcript. It doesn't encode for any proteins, but it's chopped up into small 20 base RNAs that are called microRNAs, just because they're small. And those can silence other genes, and we think those have something to do with keeping the genome from expressing and making mRNAs and proteins, okay? So that's the only thing that's made, the latency associated transcript. Okay. Reactivation. There's by the way, if you search my YouTube channel, you will find a band called Herpetic Legion. They played at a virus meeting, and the name of their concert was Reactivation.
Did I not meet somebody in this band at some point?
I don't know. I'm not sure. I don't think so, because they've never been
I feel
here.
like I've heard you have lots of weird science geek bands, though, then, right? Because I'm constantly hearing that.
Yeah, there are a lot of science geek bands, but this
Okay.
one is Reactivation. So we have now reactivation, which means the DNA begins to be transcriptionally active. You make mRNAs, they make proteins, you assemble new virus particles, they travel out of the neuron cell body, they go down anterograde transport down the nerve, and they're released just below the epithelium. The virus reproduces in them, and that gives you the cold sore, right? And so the tissue innervated by this ganglia gets the cold sore. I'll show you where that is in a minute. But not always, you don't always get blisters, okay? You don't. But if you do, you can treat them with an antiviral like, well, they used to be called Zovirax. Abreva, yeah, Abreva is the thing you can buy in those little tubes for a lot of money. That's an over-the-counter antiviral. So that's how you transmit the virus to another person. You're shedding it after this reactivation, and it's in your saliva, and you kiss someone, or you just give them your saliva in some way, and that's it. They get it if they haven't already had it. Now, you may ask,
If there's no activation, isn't there just like how do you spread it?
there has to be activation. Otherwise, the virus is gone. Yeah.
But if there's no symptoms, is there just like random times that?
Well, if you have a cold sore,
Right.
you're not going to kiss someone, right?
Right.
But if you don't and you're shedding, that's how you infect someone else.
So how do you know when you're shedding if you don't have a cold sore?
You don't. So you kiss all the time, and when you're kissing and shedding, that's when you transmit it. Okay, it's a simplistic view, but so this is how infection is transmitted, by intimate contact. Now, you may ask, how does the virus do this? We have an immune response, don't we? Well, they're slow. The immune response takes a few days to kick in, your memory, and by the time it does, the virus is made and the infection has spread. And as I said, some people reactivate every 2 to 3 weeks, some never. Some people reactivate with cold sores, some people never.
Visto Tutti is asking, is there anything that triggers or enables the reactivation?
Yeah. We're going to get to that in one more slide.
They jumped the gun.
But I want to show you where the virus goes, where the ganglia is that the virus goes to when you have an infection of your lips or your mouth. So that's the trigeminal ganglia. There's one on either side of your head, just in front of your ear, as you can see in this picture. And that is a collection of neurons from nerves that go all over your face, as you can see here, your forehead, your nose, your cheeks, your chin, your lips. So the virus, if it infects your lips, for example, it will travel down those to the trigeminal ganglia, become latent there. And then when it's reactivated, it gets delivered to one of those branches, typically the one that brought it in, right? So if you got infected on your lips, that's where your cold sore is going to be. But some people get herpes lesions on their nose, which implies that perhaps that's where your primary infection was. All right. So what is the virus is latent in the trigeminal ganglia. Upon reactivation, it travels down one of these nerves and you get a cold sore. So what is the trigger for reactivation? Stress of some kind. Like I like to tell my students, my midterms reactivate your herpes infections, your latent infections. Or sunburn. Many people go skiing and boom, they get, high altitude, lots of unfiltered sun. You get a herpes cold sore. Physical stress, emotional stress, nerve damage, hormonal imbalances, steroids, many things. A lot of people who got COVID got reactivations of their herpes, and that's not surprising at all. It's a stress, right? Any other kind of infection would do that. So those are the conditions that cause the virus to start replicating. But how does it happen? I could say UV light, but how does that happen? What's going on? Well, the first thing you have to understand is that there's a program of mRNA synthesis in a herpes virus infected cell. Things have to happen in a certain order. They don't just happen randomly. For example, Here's a timeline of a herpes simplex infection here. And when a virus infects a cell, the virus particle actually has a protein called VP16, which allows the very first mRNAs to be made. They're called immediate early or IE mRNAs. And then the proteins made from those are required for the next set of mRNAs called early mRNAs. They include the proteins needed to replicate the genome. And then finally, those proteins turn on late mRNA synthesis, and those include the structural proteins. So now you can build a virus particle and put DNA in it. But if you're looking at this, you may be asking, wait, in a neuron, we just have DNA. We don't have this VP16 protein. So how can it ever get started? Because you can't make new particles unless you first turn on that immediate early promoter. And so this was a problem that people saying it for years. How is this going to be overcome? And now we have it figured out. And that's the next and the last slide. So we're almost done, folks. Okay. This is what happens. So here's a neuron, and there's a latent herpes DNA in it, that little circle. When DNA goes in the nucleus of a cell, it's covered in proteins called histones. And covering it with histones basically shuts it down, makes it unable to be made into mRNA. So it silences the DNA. It's its natural defense of the nucleus. When any DNA comes in, it says, nope, we're shutting you down, and it gets coated with histones, all right? Furthermore, not only is it coated with histones, but the histones are chemically modified. Methyl groups are added to the histones, which further make sure that DNA is never going to be made into mRNA. So it's silent. And the cell thinks, we have this handled, right? And so here's a little picture of the DNA wrapped around these histones with some methyl modifications on them. It's completely silent. But what happens is when you're stressed, when you have your neuronal stress, your UV, you're taking an exam stress, it activates an enzyme in neurons called a neuronal stress kinase. Okay? It's an enzyme that's activated by stress, and it puts phosphates on the histones.
What?
It puts a little phosphorus, right? And that little phosphorus is enough to turn on herpes DNA transcription into mRNA. And you don't need that viral protein anymore. All the genes are turned on, and eventually you make all the viral proteins you need to replicate the DNA and to make new virus particles. So that little phosphate that's put on by that neuronal stress kinase, which is activated when you get stressed. Who knew, right? That's all you need to turn on the genome. And obviously, this is an amazingly intricate little dance here that has evolved over many tens of hundreds of thousands of years, not just in humans, but in other organisms as well. So the virus has evolved to become latent. So it's carried by a lot of people without damaging them. And then under certain conditions that we all experience, stress, mice experience stress, right? Snakes, birds, everything on Earth experiences stress. Something coming to eat you stresses you out. So
Usually
this is the system that responds to it, and it has its own reasons for existing. So neuronal stress kinases are doing something else. They're trying to mitigate the stress, most likely. But for the virus, it just is there at the right place at the right time.
Is there value to them out? Like, do they mitigate stress in some way, or can you just remove them?
Well, these kinases definitely do something to get around stress, right? So stress involves damage to proteins. And
they don't just open us up to herpes.
So
they have other value.
So these kinases are working on that, and then they inadvertently activate some virus. So that's what herpes does, folks. And I thought that would be a cool little story here at the beginning of office hours to talk about. And I hope you enjoyed it. Now let me see if I can get out of all this. I can turn off all this stuff. And you can have questions for sure. Yes, Costella has some stress. I got some stress. Yeah. Everybody has stress, right?
Yeah.
Are you stressed? Not this
Not
moment.
at all, boss.
You're cool right now?
I am so good in life. I love everything about
wait,
it.
Wait a minute. You're saying you're never stressed at all?
Never. I have the most wonderful job.
All right. Well, that's fine. But once you leave here, there are other things that happen. Right?
No, yeah, and that's when I get shingles. Generally speaking, you can tell that, and it's not just the stress. It's me attempting to repress stress for any length of time that I all of a sudden get shingles.
It's interesting that you mentioned shingles because that is another herpes virus.
Mm-hmm.
It's called varicella zoster.
Mm-hmm.
But you get it as a kid as varicella. That's chickenpox, and it remains in you forever,
Mm-hmm.
right? And then you get, when you're 50 or higher, you get reactivation and you get shingles.
27.
You got it at 27?
You have to stress yourself out pretty good to get it at 27.
Yeah, well, maybe you have other issues going on there.
Lord knows I do.
So that's herpes. I think that's a very cool story there. We know what's happening with reactivation, with latency and reactivation. A lot of people have worked on that over the years. Now, you may ask, what can we do about this? And the problem is that it's transmitted to babies at a very young age. So that's going to be, it's kind of like respiratory syncytial virus that babies get at a very young age. So maybe maternal immunization would help protect the baby. I'm not sure, but people are working on simplex vaccines. They have for years with no success. People are now trying
But the
mRNA
truth is,
vaccines.
eventually everyone will have it, so it won't be a big deal. It'll just be another function of how humans roll around on the planet,
It
right?
is. In quite a few people, they can get, on reactivation, the virus goes in your brain and you get encephalitis, and that's very bad.
Okay.
So that's in about 1 in 250,000 people who get herpes. It can go in their brain. So Troy says our microphones are good tonight. Thank you, Troy. I'm glad they sound good. So I'm on a Heil PR40 and Karen is on an SM58, right?
Mm-hmm.
It's a good sounding mic.
Yeah.
I like the sound. Maybe I should switch to it.
Well, but also we both know how to use a mic.
Well,
so
that helps.
Yeah.
Greenfield's there. He has a question. Greenfields has a question from Ireland. I hope you're well. Question: I've had one MMR at 12 years of age. Did not receive a second. Will I have enough protection from measles? Yeah, we talked about this on clinical update.
Right? Yeah.
What did Daniel say?
I don't remember. But it would have been episode
1098.
Right.
For sure. Yeah, 1098. And then we did 1099. And now his one this week is 1100. It doesn't hurt to get another dose, okay, since it's a long time ago. And measles is on the rise again. So I believe his recommendation was to talk to your doctor and get one. If anyone remembers otherwise, please let me know.
Can we throw in there, like along the same lines, John Lee was wondering why you ended up getting a COVID booster last fall?
Oh, this I've addressed before.
Right.
I originally didn't want to get it, but I have a new comorbidity, and I feel that it's important for me to have another booster. So if you're over a certain age, and I'm 71, and if you have other comorbidities, other health problems, you should get the booster. Or what you could do, so I'm going to do anyway, you don't get another booster. You just stay with your original 3 or 4, and then if you test positive, you take Paxlovid right away. I think that's what I'm going to do, should I test positive. But, I don't think you need to get a booster every year. I think the booster is going to wear off in a few months anyway, especially for older people. It makes more sense to me that you stay with your original 3-4 doses and just take Paxlovid. Yeah, that's what I recommend. Okay, let's see. Let's get some more questions here. What an interesting start to a live stream, to have a false live stream. I just freaked out when I saw it said, live stream is over.
It's ended. We just started.
Patricia loves office hours, makes my week. Oh, that's very nice. Thank you for saying that. I appreciate it. Hello, Kip from San Francisco. Well, you can get on a plane and come to New York anytime. Vic says, someone asked me if you could get a measles infection even if you are vaccinated. possibly asymptomatic. All right, so no vaccine prevents infection, right? When you first get the vaccine, you have very good protection for maybe a couple of months, and maybe you're protected from infection. But then that goes away very quickly. Then you have memory, which will respond, but it will take a few days. So you will get infected, and you may find yourself having mild to moderate disease. So if you are vaccinated, you can still acquire measles. It's a respiratory acquired virus, and so there will be a period.
Wait, Vincent, is it respiratory?
Yes, you acquire it
But is
by
it
the respiratory route.
respiratory?
Oh, is it respiratory?
Or
is
is it
respiratory?
respiratory? Or is
Gosh.
there's a British
So in the
version,
U.S.,
too.
supposedly the proper pronunciation is respiratory with the accent on the first syllable.
And
right?
then the Brits do respiratory.
Respiratory. But you call it respiring, right? And Daniel calls it respiratory.
Well, I've always said respiratory, but
And we
I'm
were
an
told
actor,
it was
not a
wrong.
scientist, so don't listen to me.
I know for most people outside the U.S., Paxlovid is not an option. But, and I will come up to Hakon's question here about HPV vaccines. You know, that people, they don't really prevent infection. But we'll get to that. Hang on. So, yes, you can get infected with measles. But here's the thing. When measles virus infects you, you inhale respiratory droplets. They do not reproduce in your epithelial cells. They enter immune cells that are patrolling your epithelial layer of your respiratory tract. And those immune cells take the virus into the blood. It takes them to lymph nodes. So you're not shedding virus right away. And so you may have time for that memory response to kick in and reduce virus replication before it is shed the other way. Because to be shed the other way, virus has to get into the blood, reproduce in lymphoid organs like lymph nodes, build to high levels in the blood, and then from the blood it goes through the bottom of the respiratory tract epithelium and back out. And that takes some time. And so their immune response may be able to kick in and prevent that shedding. It's very different from most other virus infections like SARS-CoV-2. It will infect your respiratory epithelium, and those cells will make virus and shed it right away. So because the virus can infect those cells, whereas measles doesn't infect initially the epithelium. It goes through them into the underlying tissues. And so I think the amount of shedding after a measles infection, if you're vaccinated, is minimal, if anything. But nobody really has looked at that as far as I can tell. So you probably can't transmit it as a consequence. Measles vaccination is very good at knocking down that transmission because I think of its unique pathogenesis. I have to highlight this comment by Andrew. Science is driven by experimentation, always. And you do science in your lives. When you try something and it doesn't work, and then you try something else, you change something, that's science.
Click on it.
I did.
Oh, you're before me.
Andrew, because this is an important concept. Science is driven by experimentation. And research, the means we repeat it all the time. Because it doesn't work, we want to have more repetitions and so forth. All right, so there we go. Who else is wanting to ask?
Jessica, search
Yeah.
for Jessica. She had an interesting question about bats.
Oh, that's coming below. Yeah, we'll get to Jessica. But right now we want to thank Kip, who has donated money to Microbe TV to support our science communication.
Thanks, Kip.
You know, there's a lot of science misinformation out there. People just wantonly say lies. Today on Beyond the Noise, Paul Offit was talking about the January 6th insurrection in Washington, D.C. And Del Bigtree gave a talk at that insurrection. He's a big, as Paul calls him, a virulent anti-vaxxer. And do you know what virulence is? The quantification of pathogenicity. Anyway, he said, I wish I could tell you that COVID isn't serious. I wish I could tell you that Tony Fauci cares about your health. You're all being driven off a cliff. I mean, he said COVID is not serious. 7 million people have died, Is that not serious?
Listen, the anti-vax movement and a lot of these lies, they're not driven from any real personal connection to not getting vaccines. It's driven to like control people. It's in the same way that they, remove education from the school systems in order to have malleable people that they can control. And having anti-vaccinated masses makes them probably a lot easier to control. It's a function of fascism.
That's also they want to take down the system too.
Yeah, but they don't care if we live or not.
No, they don't.
Yeah.
Anyway, thank you for your contribution. Nick Calliope says we love seeing your expression. I'm very expressionable. I make a lot of it because I don't know why I'm Italian.
Expressionistic.
Expressionistic. Expressionistic. Okay. This slide was in today's lecture. Yes, that's exactly right. Picture in picture. Now we're getting through. I have a little giggle. Do I have a little giggle? Patricia says, Vince's little giggle cracks me up. Do I have a giggle?
Okay.
I just want to know.
What's the right answer here?
There's no wrong answer.
You have what I would call a little cackle.
Like that?
Yes, it's
like
nice.
that. Yeah, that
it's
little
nice.
cackle. It's
right,
a cackle.
right.
Yeah. Oh my gosh, it's a crow. I'm actually a crow. All right, Vincent saves the day. Thank you very much. I'm glad you all. We're going to move through these comments. Technology. I got to work. I'm very. Do I use OBS? No, this is a technology question. I don't.
I'm sorry. Can I ask a question?
Yeah, of course.
Vic says they're going to. I can't pronounce that word. Mississauga this weekend for a cat show. What cat show?
I don't know. This is about viruses. I
know, but now I'm really interested in the community you've built here.
So tell Karen what cat show you're talking about.
Thank you.
No problem. Yeah, all comments are fine as long as they're civilized. Doreen says Karen got the blue glasses memo. It's very good. It's really good. Blue glasses. I'm looking for questions here. I think Markle we didn't see before is from the east end of Long Island. Oh, so Vic says there's a pet expo, which includes a cat show. All the best cats in the world will be there this weekend. Is that what you meant?
Yeah, nice.
All right, so here's Jessica's question. So bats can be persistently infected. What are contributing factors that can lead to this? Do you think their immune system attenuates virus? So what I think it is, as you know, bats fly, and flight generates, it consumes oxygen and generates byproducts of oxygen, which are toxic. And so the bat immune system has evolved to deal with those. So the bat immune system is ramped up, and it doesn't overreact. So you don't get excessive inflammation. So inflammation drives severe COVID, right? When you have real lung dysfunction, it's because you have inflammatory process going on in your lungs. Your immune system is overreacting to infection. And so bats have learned how to control that through years of flight, right? You have to be able to control inflammation. And so viruses trigger inflammation, and a lot of the disease symptoms and signs of a virus infection are caused by inflammation. And so bats have a way of dampening inflammation. So a virus infection does not cause inflammation and hence doesn't damage the bats. I mean, not all virus infections spare bats, right? Bats get sick from some viruses, but many of them don't because they don't trigger this inflammatory process, which is really the killer in many virus infections. So those viruses can remain in the bats for extended periods of time because they cause no harm. They don't kill the bat, and the bats can live fruitful. They can live long and prosper with these viruses in them. So that's what I think, and that's what a lot of people think. Jessica, I don't think it's a matter of attenuating the virus, because we can get one of those bat viruses and it wreaks havoc in us because it's been in a bat for millions of years or tens of thousands of years, and they've gotten to know each other, and it's in us. And so, whoa, where am I? Let me out of here. And it causes collateral damage. So that's what I think is going on. F. Yang says, herpes 1 was the first virus I presented in my virology class back in undergrad, November 2019. Yeah, it brings back memory. By the way, speaking of memory, listen to this story. I went to dinner with a student in my class last night, Davey, and he. So it was an event for faculty and students to have dinner at Columbia, and he told me in high school he was listening to my podcasts.
Oh, yeah?
And now he's having dinner with me. So it's like, wow, he's taking my course and he's having dinner. I said, it's no big deal, but it's really nice to meet you, right?
Nice.
It's cool that you can. He said, I found you in high school and I listened to your lectures and now I'm sitting in on those very same lectures. That's great.
That's awesome. Boy, you're getting them early, eh?
I want them really early. So here's Wayne's comment. Epi means around or near, and some means body. Perfect. Thank you very much.
What's the word again?
Episome.
Episome. So, am I in some reference of episome to you?
Yeah, you're an episome at the incubator. You just are around the body of the incubator. So it could be used for that. But the
Okay,
word.
so we could actually use it in day-to-day language if we chose to.
Right, you could.
Because you said, yeah.
Yep.
I'm just going to start using it all the time.
And Visto says, herpes loves us all, silently loving our nucleus.
Mm-hmm.
So did you quit ECAM? Is that what you did? So that must have triggered the shutdown, right?
Mm-hmm.
Wow. So it doesn't matter where you're quitting from. If mine's still running, they say, oh,
I think so.
interesting.
And I think that I just was under the impression when we did Tweevo in Johnson
We
City
both
that,
had it,
that
yeah,
you
but
were
I
on
didn't.
ECAM. I
I
didn't
wasn't.
realize you weren't. Yeah.
I was on what you're on now.
Got
the
it.
stream.
Yeah.
So this is funny. There's a little. So Steph said, "I thought you meant blue light blocking glasses," when she said blue glasses. And Doreen says, "I did think blue light glasses too, because we're all science geeks. That's why you guys are my peeps."
Right.
Can we use? B wants to know: can we use Clovirax? Clovirax. It's a cyclovir. Yes. It's an antiviral for herpes virus. You can use Clovirax for sure. Yep. Tom says, would a vaccine analogous to Shingrix be feasible to suppress, reduce reactivated outbreaks? That's a good question because, look, we're all immune to herpes viruses because we've been infected. And even with that, by the time the memory kicks in, the virus has reproduced and spread. So I don't know that a vaccine would do much better than that, Tom, than natural infection, right? I think the best thing you can do is prevent infection. Well, you don't prevent infection, right? You're always going to get infected. That's the problem because you can't prevent the infection in the mucosal layers with herpes simplex. So once the virus gets in, it's going to go down the nerve into the ganglia and it's slipped away from you. So I think it's a very tough problem. Same thing with HIV, right? If you don't get that virus within the first 2 weeks that it infects you, it establishes the latent reservoir and you're stuck with it forever. So that's very tricky to do that. So because vaccines do not prevent infections. And so that's what we're up against. Here's a good question. Do the herpes virions damage the neurons as they travel and exit to the mucosal cells? I used to ask that question all the time of Saul Silverstein because you would think that the neurons are going to be damaged. And he said, no, they are not damaged by the exit of the virus. So the viruses reproduce in the nucleus. They bud out and then they flow down the nerve fiber, the anterograde flow to the skin or the mucosal surface, and they bud from that so they don't damage the cells. No. John has a one-man band called 1PFU.
Care to explain that to me?
So PFU is a plaque-forming unit. It's a measurement of virus infectivity.
I knew it was funny. It just was a little over my head.
That would be a good T-shirt, don't you think? Do you think it's funny?
Well, now that you explained it.
Yeah, 1PFU, but it's an insider joke. Not everyone's going to get it.
Totally. That's why I asked for an explanation.
Yeah. A good instructor will lead you to question what they will be answering soon.
Yeah, that was actually a brilliant comment made right at the moment when
When
I
was
asked
that?
you a question that you were just about to answer.
The good instructor will lead you to question what they will be answering. Is that right?
Yeah.
I never heard that, but it makes sense, right? Because you're planting ideas that make you think and ask questions, and
Yeah,
they're the logical.
I was trying to give somebody voice in real time and you were like, I'm not there yet.
Yeah. Cool. Thank you, Vic. That's a good one. Blue light, blue frames. So Vanity doesn't remember me saying that in class, that the exam itself would cause reactivation. Yeah. So you took the class online, Vanity, and maybe I just didn't say it then, but I said it today. I
I was
gave
going to
this
say,
lecture today, and I said,
it could be new comedy for you.
I have a few things that are good. I think the gastroenteritis thing is really funny. I think, unlike love, herpes is forever. This exam will cause you reactivation. Yeah, there are a lot of good things. It's not hard to make science funny, you know?
Well, not when you're talking about STDs, because God knows those are just hilarious.
John says phosphorus seems important in a lot of biochemical reactions. Isn't it also key in cell entry? Yes, well, adding phosphates or phosphorus, we actually call it phosphates because it's in the form of a PO4, a phosphorus with four oxygens, right? They can be added to proteins to modify their activity, as we saw with the neuronal stress kinase. It added a phosphorus to the histones, and it activated the herpes genome. Many, many other reactions. And so those proteins are called kinases. Protein kinases. They put a phosphorus onto a protein. And then you ask, is it also key in cell entry? Well, as you know, the membrane of a cell is a phospholipid bilayer. It's composed of phospholipids, which have a head, a phosphorus at the head, and then a fatty acid tail. It's called a phospholipid. The phosphorus faces out. It's charged, so it keeps things out of the cell. And then for things to enter, they have to get around that. So, yes, it plays a role. And so making vesicles for cell entry is all dealing with those phosphates, yeah. Steph says shelter cats get ocular herpes from the stress of being in the shelter. Not surprised. Very stressed. You know what else they get? They get coronavirus, that transmogrification of the corona into something pathogenic from the stress of being in a shelter as well. Meg wants to know if shingles goes through a similar thing. Yes, except shingles, the virus of shingles is varicella zoster, so you get it as a kid as chickenpox, and the virus goes latent in nerves beneath your skin, all over your body, so not in the ganglia of your head or genital area. So these can go under you. So you get chickenpox all over you, and then the virus can go latent beneath the skin and nerves there. And then when you get reactivated, and we don't know the conditions that cause stress, that cause varicella zoster to be reactivated, then the virus will bubble up on your skin and make a nice line of vesicles in one line, perfectly aligned with the nerves that innervate that skin. It's called a dermatome. Okay? Do you know that word, Karen?
I think it sounds like a color.
An area on your body that relies on a specific spinal nerve. So one nerve, dermatome.
But that's not the name of the nerve.
No, it's just a general name for an area of your body that's controlled by one nerve, a sensory nerve.
So why does, if it's the same freaking virus, why does it present differently than chickenpox later on because you're old? Why does it look more like tapioca pudding?
Well, it's still
bites?
vesicles, right? It's kind of this similar thing. It's just in one area. They're all concentrated in one area, whereas the others will spread all over.
I also don't remember chickenpox giving me like the nerve pain.
Well, it's different in everyone, right?
Yeah?
Mm-hmm. Well, the chickenpox doesn't give you the nerve pain. So,
Okay.
I mean, the shingles can give you nerve pain because it's coming from the nerves, right? and it's probably damaging them somewhat. But chickenpox is not doing that. It's going from the blood to your skin, and then from the skin, it's going
So
into
it's
the
about where
sensory
the
nerves,
virus is living once it's reactivated?
where it's reproducing. Yeah.
Okay.
Mm-hmm. Roy says, are there antivirals against these viruses? Something that I'm missing. Moderna had a big focus on EBV, HSV, cytomegalovirus, VSV today, by the way. Okay, so we have really good antivirals against herpes simplex viruses that also work for EBV and cytomegalovirus. And there are only a few because they're really good, and any company that develops a new one, they can't break into the market. They just can't, so they don't go anywhere. But the ones that we have are good, and there is some resistance, but not enough to warrant having others. So that's the story. Kathleen says, your assistant asked some good questions to help us non-scientists. Thank you.
That's what I'm here
That's
for.
That's Karen, the incubator regulator. What animals other than humans get herpes? Oh, all kinds of animals. I know there are reptile herpes. You know, the dinosaurs had herpes. There are fish herpes viruses. I'm sure they all get herpes. Let's look. Which animals get herpes?
I love it when you Google during this.
Insects. Well, that's the thing. I'm not going to have it all in my head.
Of course not.
We have a wonderful lady from New Orleans who once said to me, Vincent, there's two kinds of information. The kinds that you keep in your head and the kinds you know where to look for.
Right.
You don't have to keep it all in your head,
No.
right? All right, so herpes viruses have been found in insects, fish, reptiles, amphibians, and mollusks, as well in every species of bird and mammal that has been looked at.
So herpes has been here longer than us.
Oh, much longer than human beings, for sure.
Yeah.
The dinosaurs had herpes.
So basically, herpes has more of a right than we do to this planet.
Yeah,
is
but
what you're saying.
they're not sentient, so they don't get to do anything. It's a good question, Nova guy. I like that. All right, so F. Yang says, I saw in a paper that frequent reactivation of HSV1 in mice leads to higher Alzheimer risk. If the virus is not in the brain, how does that work? This is a really good question. So there is some association between herpes simplex and Alzheimer's, for sure. And I always said, well, what is this about? And you can see, the idea is that you're getting reactivations in the brain which are not pathogenic, but which lead to plaque formation and eventually Alzheimer's, right? And I always thought that whenever herpes went in the brain, it's a disaster, right? You get encephalitis. But apparently it goes in more often than we know, and it's not pathogenic. It gets reactivated, and then the formation of plaques in your brain is a kind of an immune response to restrict whatever process is going on there, in this case, a virus infection. And so, yeah, there are currently some clinical trials ongoing to treat people with like 4 grams of acyclovir a day, people who are at early onset Alzheimer's, and treat them for 2 years with 4 grams of acyclovir a day and see if it makes a difference in their outcome. I don't know if it will, and I don't think it's going to be everyone, but it could be a contributor, yeah.
Wow.
Mm-hmm. I thought that you had herpes in your brain. That's it. You get encephalitis, but apparently not. Apparently not. All right, so James says his wife got it at 31 in her eye. It could be herpes or shingles. I'm not sure maybe herpes, right?
I'm pretty sure it's shingles because he's probably not going to rat out his wife having herpes.
Well, everybody has herpes.
I know, but...
Listen, here's Carol. So, folks, if you tell your medical histories here, this is a public forum. Okay?
Right.
So I'm assuming that you're okay with us talking about them. So here's Carol, Dr. Wong. I was on vacation in Costa Rica. It was so relaxing until our return flight got canceled and rebooked the next morning. I got a cold sore. Yes. You got stressed out. Your neuronal stress kinase was activated.
I mean, I think societally, simplex 1, because it's generally in the mouth area, is pretty passe. Nobody has any real issues with it anymore in 2024. Simplex 2, because we still have some kind of puritanical ideas about things that are actual sexually transmitted diseases,
Mm-hmm.
not just through intimate contact. I think there's a little more paranoia about who has
Yeah.
that data.
James said his wife had shingles in her eye.
In her eye?
That's what he said. I mean, it can't be the eyeball. It must be the
Ow.
tissues around the eye. But 'cause
Ouch.
the virus is not being latent there. It has to
Oh,
be in
my
the skin.
goodness.
But that would be very painful.
I really have
Yeah.
eyeball issues anyway.
Can humans get herpes in the eyes? Absolutely. They. Now that I think of it, yeah. So she could have gotten shingles in the eye. So there's an infection of the eye called herpes stromal keratitis. So the virus infects the outer layer of your cornea, the epithelial layer, and then the underlying cornea gets an immune response that makes it opaque. So you get multiple herpes infections over the years. They get more and more opaque until you're blind. This is the leading cause of blindness in what do we call them? First world nations? Is that the right word?
Okay.
What is the word?
It's probably not.
It's not developed nations, right?
Right.
Economically,
There's nothing we can say here without Googling that isn't going to make us sound like
you know what I'm talking
colonialists,
about.
But I do.
In places like the U.S., it's the most common cause of
Now,
blindness.
technically, in the old way of talking about U.S. is first world, but I don't know that we do first world and third world anymore because I think that's like colonialist language. But I don't know. I'll have to look it up.
Okay. Anyway, so yes, so I suppose that it did infect James's wife's eye. Yeah. This is funny. Lise says, "I have given up on stress." How do you give up? You can't really control it, can you? I don't think you can.
Well, apparently they did.
Well, maybe if you meditate and
Just decide you're not going to play with that anymore.
decide. Thank you, Abdulaziz, for your contribution to science communication. We really appreciate your support in trying to get the real message out there. Can varicella zoster reactivate earlier than 27? Yes. I knew a 21-year-old who once had shingles because that student was taking immunosuppressive drugs. So I think that the reason you get it 50 and up is because you slowly become less immune competent, right? You undergo immunosenescence as you age. And so if you're artificially immunosuppressed, younger, boom, that's going to do it. Can I highlight a political statement by someone? We should stay away from politics.
Do whatever you want. It's your show. What's it say?
I get stressed every day. Every time I hear former President Donald Duck.
Oh my goodness! I don't know what's happening.
Your phone is playing.
I don't know why. Oh no. Okay. I was looking up what language you use. They get stressed every time, and I know how that ended.
Yeah, they hear former President Donald Duck. I didn't highlight it, though. Daniel says, do we know why some people are unable to produce herpes simplex antibodies? I don't know the specific reason. Many people, when you immunize 100 people, not all respond to your immunogen. They don't make antibodies or T cells, so they have some defect in either sensing or evolving. So they have a defect in their immune system, but it's not just specific for one pathogen. And I don't know if that we understand why it is for herpes, right? But it certainly happens with all viruses.
Okay, so I have an answer to how do we reference countries that have healthcare systems that still have work to do and such things. And I think it's pretty consistent. It looks like first, second, third world sounds hierarchical and as if one is better than the other inherently, and people aren't using that anymore. Developing countries is somewhat acceptable, but it looks like there's other resources of ways to explain it. I think you're safe if you say developing countries, and then I'll continue to find you better language later.
YZ says, can TWIM audio only resume being put on YouTube? It stopped at $2.99. Yeah, I used to take the TWIM audio and just make a video track with just the title, the whole track, so people could listen to it on YouTube.
Right.
And I stopped doing that.
Why?
I just ran out of time.
Yeah, so that's going on my list? Did somebody like it? Is that what's happening?
It's not hard to do.
Okay.
Someone-
It's really not.
Rob wants to know who's talking. That's Karen, the studio regulator.
Hello, Rob.
Who's sitting here. We're experimenting with her being here as kind of, what would you call yourself, Robin?
That was your reference, but the non-scientist foil, maybe?
Yeah, she just
Yeah.
flags things I might miss and maybe asks
Exactly.
questions to clarify things. I hope you like it. I think it's a good idea. So we're up to 200 people. That's about normal now. So the failed intro didn't hurt our stream tonight.
It's not the size of the audience that matters, Vincent.
Karen keeps telling me it's not the size of the audience that matters, but I like having a bunch of people here. I don't need 1,000 people like we used to have. And you know what? I do this for 10 people.
Exactly.
Would I do it for one person?
Well, you do it for me all the time, just wandering around.
Then
the
I would do it
office.
for one
Yes,
person.
You would.
You and I could have a great conversation.
We could.
But so let's say Lise, whose name is right in front of me, and I were the only ones on this stream. Lise, would you talk? Would you ask questions? Or would you then wander off into something other than viruses? What would you do? Any of you, if you were just stuck here with me and I said, yeah, let's do the stream with just you, would you be able to do it? Well, you don't have to drive it. I would drive it, of course. Yes, Will. So this is a problem for most people outside the U.S. Paxlovid is not an option. I recognize that. I think it's unfortunate. It shouldn't be that way. So yeah, you're going to have to get vaccinated every year, most likely. Here's Neva from Buda. Remember Neva? Did you meet Neva in Johnson City? Sure you did.
Yes, I did.
She's here. The Tweevo in Johnson City was great. The Science Mill is a wonderful establishment, isn't it? Great people. So good to see you. Good to see you, Neva. Thank you for coming. Okay. Håkon. Is this the same Håkon as from TWIP, do you think?
I don't know. Ask.
Are you the same?
How's it spelled?
Hakon.
No, there's that.
Different accent?
Are there is there like the two little dots over the O?
No, of the A, you mean?
I don't know.
Anyway, Håkon.
Håkon is how it's
Håkon.
pronounced.
Gardasil 9 seems to provide protection against infection with HPV. You know what the problem is, Håkon? You can't measure HPV infectivity, so you actually can't do the experiment. It is 100% effective at preventing cancer, no question. It's a great vaccine, but I'm not sure you can say it prevents infection because we have an infectivity assay. All we do is PCR, and that's just not good enough unless there's zero PCR activity, which I doubt. Yeah. Privyet, very Nika, Veronica. Privyet. I'm practicing my Russian. Privyet. How's that sports nutrition science podcast with Mingarelli going? Any movement? It's moving. We're developing it. It's going to happen. Would you say it's going to happen?
Well, I can say, just from my perspective, they have headshots taken. They're working on a logo. I mean, all the important stuff is happening, and yeah, we're just waiting to hear back from them that they're ready with structure, and then we'll implement. We'll totally give you guys a heads up. You'll know before anybody else does when it comes out.
Veronica, I know I have to get you the lecture transcripts for you to translate into Russian. I'm just a bit swamped with the course right now, but I will do that because Veronica is going to translate my lectures into Russian. Very exciting, and have someone else doing it in Spanish. It's going to be a lot of fun. Reach more people. This is great from Richard. Respiratory with an EAR versus respiratory with an EYE. Just different face parts. You got it, ear and eye. That's great. And Doreen says to Karen, oh, that makes sense. I'm an actor, you said. I was thinking dialect coach before you said you're an actor. Cool. And then she also says, it's a small world I usher at Hartford Stage.
Yeah, I did. She said she's from Elgin, and I actually did my Janis Joplin cabaret show years ago at like Elgin Community College or something there. They called me from Elgin and said, hey, come do the show. And I had my Chicago band play at it. It was a great time.
Where is it? In Illinois?
It's in Illinois, yeah. It's not far from Chicago.
Cool. It sounds like a new EUA for a COVID monoclonal. Will it be covered with Daniel Friday? Yeah, I'm sure. I'm sure Daniel will talk about that.
Do you want me to write it down?
Yeah, why not?
All right.
Why do it have to be an emergency use? So, yeah, the FDA grants EUA to monoclonal antibody for pre-exposure prophylaxis of COVID in immunocompromised people. So that's a monoclonal called Pemivibart or Pemgarda by Invivid, and it's for adults to adolescents with moderate to severe immune compromised conditions. Yeah, I'm sure he'll talk about it. Hat says Dr. Griffin recommended a full series of 2 shots of MMR vaccine. Okay, thank you. So if you've only had one, get another one, according to Daniel. Thermodynamics says the internet is a very mixed blessing.
That it is.
Every blessing is mixed, isn't it?
No.
I mean, whatever is good, there's going to be a dark side of it.
That was a little dark, Vincent.
I'm not being morose or anything. It's just the way it is, right?
Yeah,
It's just the way it is. Sean says, virus reverse genetic systems. I've seen some researchers use apart from TIV12A for adding reporters. Why and how does it work? I don't know what TIV is, but I know 2A is a sequence from some coronaviruses. So what it does is it's a ribosome stop-start signal. So let's say you have a viral gene, then you want to put a reporter next to it. You want them made separately. You put a 2A sequence in between. The ribosome hits the 2A, it stops, the previous polypeptide is released, and then it continues translating your reporter gene, for example. So it's a great way. It's a very short sequence. It's NPGP. NPGP, those are 4 amino acids. And you put that in, you can get 2 separate polypeptides made. It's just amazing. It's just amazing. Yeah, that's 2A. I know that for sure. Angry Penguin says, many physicians say wait and see before prescribing Paxlovid. My 80-year-old mom with B-cell lymphoma was told by her family doctor on the second day of COVID to take Tylenol. Yeah, that's not going to help, right? It's going to relieve some symptoms, but the virus is going to rage on, and then that person, 80 years old, needs Paxlovid. So hope it all worked out, Angry Penguin. Oh, then we go. Next day, she talked to her oncologist who immediately prescribed Paxlovid. Okay.
There we go.
Good ending. Very good.
Yes.
Because we don't have enough. We only have 140 characters here, and so she had to split it up. Excellent.
I was a little worried there for the first half.
I was
though, right?
worried, yeah, because Tylenol, and I was afraid that she was going to have a bad outcome, but good. Siddhartha, thank you for your contribution to science communication. So Siddhartha was at TWIV 1000.
Okay.
He lives in New York, and he's a computer programmer, and he offered his services if we ever need programming.
Okay, great.
Okay,
Well,
so
Siddhartha, I was at TWIV 1000. I was the bartender.
no, you were the manager of the bartenders. That's what you tell
That's
me
what
all
I mean.
the
The
time.
bartender lead. Like I can't remember the word for it because I haven't done it in so long now that I've been here. I probably served you wine, Siddhartha. That's all I'm saying.
He served me wine, that's for sure.
Mm-hmm.
It was sent to me across the room. Hogger51 says the USDA has confirmed highly pathogenic avian influenza. That's right, in 2 dairy cattle herds in Texas. Correct. Does pasteurization kill and activate such virus particles as claimed by the dairy industry? Absolutely. Absolutely will inactivate influenza virus. This is not surprising because birds are widely infected with H5N1, and especially birds that fly over long distances, and they take a break at a cow pasture and drop off some virus, and that's how it happens for sure. So the cows got infected, yeah. Since we know so much, Raise 11 says, since we know so much of COVID happens in epithelial cells, why wouldn't we want to come up with a treatment delivered via nasal spray? You would like to, but it is short-lived, right? If you have just like a inhibitor, if you spray it on the nasal mucosa, it diffuses away very rapidly. Whereas it's something that you take by mouth, you can take a pill every day for so many days. You maintain a certain systemic level of the drug, and that's why it works well. All right, so a nasal spray of an antiviral. I mean, there is an antiviral nasal spray for influenza, right? It's Relenza, and it's not very good. But the other one, which is taken by mouth, is also not very good. So that kind of inhibitor just doesn't work well against influenza virus. I would be skeptical because of the short-lived nature, unless you do multiple nasal sprays. So those are the shortcomings I see. There's very few development in this area by pharmaceutical companies. So I suspect that there's some fundamental underlying issues that's preventing development. That might be part of it, or it could be something else. So it's a good question. Yeah. Thank you, Loretta, for your contribution to science communication. I'm still chuckling that the stream ended after 20 seconds. I can't stop thinking about it. It's so funny. Thank you, Loretta. We appreciate your contribution to make science, real science, to talk about real science here. That's what we want to do. Very Nika says, I just wanted to share that recently I joined a friend of mine, a science journalist, in a lecture about misconceptions about medications and dangers of alt med in Russian. Science communication is the dream. Hey, did you hear that? Science communication is the dream.
It's a big deal right now.
And we've been doing it for 15 years.
I know. You are part of like the old guard that initiated this.
She
but I'm
just
saying,
called me an old guy.
Guard. I said guard.
That's fine.
We're early on in the establishment, but it really is like every conference that we go to has some kind of workshop on science communication. It's something that people are actively paying attention to as an important way to move forward. That they also all have like diversity, equity, inclusion workshops of some sort. I mean, there's all of these, and science communication is right up there with the ways in which people are starting to look at the world, I think, and it's kind of exciting.
Well, when I started, I was just interested in letting people hear what we were saying, right? Oops, you just pulled the plug out. Oh, it's back in.
No, I didn't. I'm nowhere near a plug.
You're nowhere near a plug?
I don't think so.
Yeah, okay, but the monitor went out briefly.
It happened at the exact same moment that I sat up in my chair.
Static electricity? Don't do it again.
Okay. I won't move anymore.
Anyway, Veronica, let me know if we can help in any way. I think having the lectures in Russian is a big help, but maybe something else is good too. Okay, James writes, I have a non-science question for Vincent. We are always happy to get.
Did you thank Loretta?
I did thank.
Did I miss it when I went to go get a phone cord? Okay, just check.
I did thank Loretta. Yeah, thank you.
Excellent.
James says, I have a non-science question. When using your home office, I see a Fender Stratocaster behind you. It's not a Stratocaster, actually. It's a Fender, but it's one of those, what is it called? It's $200. A Strat is thousands, but it's the poor man's Fender. I think it's called an Aircaster, okay? It's not very well made, but it's red, and it looks like a Strat. I used to play years ago. I don't play anymore. It's been 15 years, 20 years since I've played.
Wait a minute.
And I'm not playing anymore.
You used to play?
I can't play anymore. I lost it all.
You used to play. I did not know this.
There's a lot you don't know about me.
There is a lot. And also, maybe it would be fun for us to, I don't know, fundraising auction off you playing a song for people or something.
I could practice a lot.
Right?
Yeah. Yeah, so I used to, and also James says, I was lead guitar for our church band for 18 years. That's cool. I don't play much at all. Where are you, James? We should get together, right? Here's Germalina Lacorte from Davao City, Philippines. Well, we sometimes have people from the Philippines, so welcome. That's pretty cool. I don't know what time it is there. It's probably, I bet it's morning, right? Philippines. I bet it's morning. Let's see. Do we know, John says, do we know why animal viruses are always enveloped? No, they're not, John. They're not always enveloped. We have polioviruses, which are not enveloped, right? Noroviruses, caliciviruses, adenoviruses, many of them are not enveloped. So sorry about that. Emily asks, I was wondering if your students fanboy when they meet you.
Yes. Can I answer that? Yes.
I wouldn't know. You should tell.
Fanboy, fangirl. I mean,
they do?
I'm not necessarily talking your students, but there are young people that are very excited to take selfies with Vincent wherever we go. And it's part of one of the joys of my existence to help facilitate that.
So the students are very happy that I'm teaching this class, right? And before class, there's another class in the room. So we wait outside, and 5, 6, 10 people that we have a little conversation, and they love it. They come and they smile and say, "Hello, professor." And it doesn't happen with all
Well,
professors.
I went to the first
First
lecture,
one.
the very first lecture of this year, right, that you had, where you
did.
introduced yourself and everyone, and there was already a Microbe TV bumper sticker on somebody's laptop. And then at the end of the class, he offered for people to come talk to him, make himself accessible. And literally, there was a line of 20 students almost immediately standing by the desk waiting for a chance to talk to him. So I'm just saying, it's super cute.
I appreciate that. I mean, I appreciate that people like it. Okay. It just makes it easier for me to reach out and teach people when they're receptive. And as I said, that I ate an orange before class in the front of the room, and one of my students said, "Why did you eat an orange?" And I said, "Well, they're good," and
right.
my assistant told me it would make me look more human.
I'm constantly going, "Oh, go ahead and do that, Vincent. It will make you appear human."
And she said, "Well, professor, I think you're human already." I said, "No, but the point is to be more accessible and not have this barrier, because there's an automatic barrier between professor and student. I don't want that barrier. I want you to feel free to ask questions anytime." Because a lot of people don't ask questions because they're afraid, right?
And sometimes there's an automatic barrier just between like scientist and, normie.
Yeah.
Like regular people who don't know enough about science, because we've all, placed people beyond us in some way, right? They know more. They must be in some hierarchy away from us. So everything that you do that makes you more human, like eating and
eating. Well, making mistakes and saying, I was
mistakes.
wrong.
Looking things up on Google,
I don't know. I don't remember everything, but I know when I see something, if it has the right smell.
Right.
I can tell if it's fake or not.
Right.
Vanity Nutrition, you don't have a Microbe TV sticker? How is that possible?
Oh, no.
I thought, I guess when you were visiting, it was before we had stickers. Of course. Well, I will send you some. Just send me your address, please, Vanity. I may have it, actually, but send me where you want me to send it
Actually, let me just throw this out here now out loud. If anybody wants to send an email to incubator@microbe.tv with their snail mail address, we shall send you stickers.
Yep. Incubator@microbe.tv. Older and Wiser asks, do the chemo treatments for B-cell lymphoma erase memory cells like measles? I don't know about the chemo, but, there are monoclonal antibodies for treating B-cell lymphomas, right? Like rituximab. And those can affect memory B cells as well as the effector B cells. But I don't know about chemo. That I don't know. That we'd have to give to Daniel Griffin. Vic loved immune this week. Yeah, we did. We each did a paper as a snippet, 15 minutes each, and that worked really well because you don't get too deep into the weeds, and then you get an overview. So one of the papers was the guy in Germany who got 217 COVID vaccinations.
Oh, that was real?
Yeah, it's real.
Oh, my goodness.
He gamed the system, and some days he got 2 a day.
What was he hoping to achieve?
He was deathly afraid of getting COVID.
So he was just hoarding vaccines when they were
sparse?
sparse?
He just went and got them.
Okay. It's not when you had to wait in line to get one. It was like anybody could
it was
go.
like last year.
Wow.
And so he never got COVID, but his immunity is not much better than people who get 3 or 4 doses. So you don't need all that. It doesn't help you anymore, right?
But did it help him psychologically be less afraid?
I suppose it did. Yeah. I suppose it did. So we will keep that up. Angry Penguin says, the mother is good. She got over COVID quickly. Thanks. Yes.
Excellent.
All right, here's an interesting one. Bree Star says, are there any viruses that infect humans that are considered helpful or good? I remember you saying that there are many viruses that infect humans but do not cause disease. Thank you. So, yes, we're full of viruses, at least a dozen. These latent viruses, the herpes viruses, they're off most of the time, but they're periodically on, and we're okay. Maybe that's beneficial in some way. And then there's the other viruses that reproduce continuously. The anelloviruses, for example, and the polyomaviruses. They are always with us. We're excreting them all the time. And I tell my students, you can excrete up to 100,000 plaque-forming units per milliliter of urine of these viruses. So public toilets are full of everyone's polyomaviruses. And when you flush them, they aerosolize them. Anyway, are they beneficial? Many of us think they are. Like our microbiome is beneficial, right? But we can't do experiments with viruses to know that. We have broad spectrum antibiotics, which we use to wipe out bacteria for surgery, and and we don't have the same for viruses to know if you wiped out your virome, what would it do? And by the way, every tissue in your body has some kind of virus in it, from your fingernails to your skin, your eyes, your respiratory, digestive, genital tracts, urogenital tract, your brain.
Okay, so is it respiratory?
What did I say? Did I say respiratory?
No, you said respiratory.
Yeah, that's the way I've always said it, probably, respiratory. So I think there are some, and there are certainly some experiments in animals that show that viruses can be beneficial to, like, mice and insects and plants, but we don't know for sure in humans. Yeah. So John says, one PFU hasn't gone viral, needs more band members. Very good.
So, wait, I think that was an invitation for you to practice the guitar.
Yeah, but I can't. I'm too busy. I can't do it. I can't do anything. I can barely do. Come on, Karen. You're the one who complains. I can't do everything that I'm trying to do.
That's true. That's true.
So don't give me more stuff.
Though I do think adding some, music or art or something in is never a bad idea.
I agree. It's probably good for my soul, right?
Exactly.
Steph's comment about ocular herpes in cats with stress of shelters was relevant. Are the cat convo. Sprinkling lysine in the diet gets rid of it effectively, so say the vets. Okay, I wouldn't know about that.
What's interesting is, like I said, my mom gave me herpes when I was very young. I've never had a cold sore, but lysine was part of our regular diet in pill form. And they claimed it was for that.
Look,
so
Andrew says I should do science stand-up.
Has he not seen your stand-up?
I'm sure he has.
Andrew.
I think Andrew's being sarcastic.
No.
Yes, he is.
No.
He is being sarcastic. No. Yes.
Vincent, think about it.
I'm thinking.
How bad could science stand-up be? I mean, you've met scientists. It could be terrible. You would do it great.
I'd need some practice.
Andrew's saying that there is a market for science stand-up, and we should put that on the list.
Put it on the list, and you help me come up with some new science jokes, okay?
I'll just write down when you're funny.
Oh, boy. Okay. Anyway, it's on YouTube, my 5 minutes of science stand-up.
And we did it in Germany.
Vanity says, of course I remember the unlike love line. I've even quoted you numerous times. Okay. Greenfield says, a company called EcoHydra claims to have a non-alcohol hand sanitizer that kills norovirus. How might this be possible given that hand sanitizer typically can't dismantle capsid protein? Well, I'm surprised because the noroviruses, of course, pass through your stomach, which is low pH, and then there are detergents beyond it, and there's high pH, bile salts, and it's stable to all that. So the only thing I can think of would be a small molecule that inserts itself into the capsid of the virus and disrupts it. And that would be
the only
feasible.
non-alcohol I've seen that say that are ones that are hemp-based. Don't know if that has anything to do with it, but I have seen like hemp-based hand sanitizer type stuff that says that it kills it.
Andrew says, Karen has got my meaning about the comedy. You got my meaning,
Yeah.
he says. You got it?
Yeah.
All right. So I don't know. I think it's feasible, but we'll see, right? Okay, Tom says, got it. Preventing a primary infection is a no-go, but my question is secondary outbreaks, by analogy to chickenpox, reduced by Shingrix. So I suppose if you had enough antibody or T cells at the epithelial barrier, you could reduce the symptoms, the cold sore, right? But, people have tried many herpes vaccines and they haven't worked so far. And so maybe there's a fundamental flaw in trying to do that. But I understand now what you're saying. Yeah, just like Shingrix, where you've got the virus in you, but when it reactivates, the Shingrix tamps it all up. The Shingrix-induced immunity blocks all that virus so you don't get cold. You don't get the zoster. Yeah. I don't see why not, because it's release of virus from a nerve ending, right? And so it's the same thing happened in the trigeminal ganglia. So in theory, it should work. But you have to have the right immunogen, right? So that's the thing. And Doreen asks the same question. Well, how does Shingrix work, and why can't something like it work with herpes, right? So they're similar viruses, and as we just said, you catch the virus as it's coming out of the nerves. I don't think the antibodies are going in the nerves and disrupting the virus there. I think it catches them when they come out and neutralizes their infectivity so they don't enter the epithelial cells. So, yeah, I think that's a good one. Yeah. All right, so Steph has put up the link to my stand-up. Thank you very much, Steph.
Good work, Steph.
Wayne says, if you're interested in phosphorus, check out The Devil's Element by Dan Egan. Wow. Do you know that book?
No.
Write it down, please, Karen.
Okay.
Now I have a replacement for sticky notes.
See? I am just one ginormous sticky note.
Okay, Larry says, if vaccines can never prevent infection, how can they ever eradicate disease? Well, you can't. So you have a virus that causes an acute infection, so it comes and goes. And you vaccinate to limit. So it doesn't prevent infection, but it severely reduces infection and it reduces transmission. So the amount of virus you need to really percolate through a community is reduced. And so eventually, if you vaccinate enough people, you don't have any more transmission. Alan says that Paul Offit pointed out recently that the incubation period is significant. Long incubation allows time for B and T cells to wake up, no illness. Short lets you get mild illness. Exactly, right? Viruses with a longer incubation period are much easier to control with memory responses. But remember, during the incubation period, viruses are replicating and they can be shed. So you can still get transmission until that memory response kicks in. Doreen says she got shingles in her mouth 2 weeks ago. I'm sorry. And we're talking about it tonight. How about that? You feel a ringing in your ears for that? Okay. Cats get herpes. We're talking about shingles now. Steph says you're funny, Karen.
Oh, thank you, Steph.
I have yet to hear anything funny out of you. Is that funny? My God, she's laughing like crazy.
That was good.
Richard says, I've heard that Einstein never bothered to memorize his phone number. You can look that up.
That is in reference to, do you want me to tell you? That you don't have to remember everything. You can look some things up.
Yeah,
Remember you were talking about
your
that.
phone numbers now, you
was a
just keep
callback.
on your phone. You just speed dial, so you don't have to remember
But
anything.
how you were saying you can't remember everything.
Yeah,
and
yeah.
now you're being equated to Einstein.
Well, that's cool. I like
Right?
Albert. He was a smart guy. He's a great. Doreen says you have a great laugh. Okay. Alina. So let me give. I'm going to pass this problem to Karen. So Alina is from Coxsackie, New York, and months ago she was saying, I need to buy a cheap microscope, and my father's microscope that he used in his practice was sitting next to me at home. I said, you can have my dad's. It's really heavy though. I need to pack it up in a wood crate and ship it. How can I do that?
Where is Coxsackie?
Upstate New York. It's what Coxsackie virus was named after.
Ah.
It's exit 21 on the New York State Thruway.
Do we ever go in that direction?
I've been in that. I've been to Albany where there are virologists. So,
But I mean, is there any, like, because that would make the most sense other than shipping. Otherwise,
I told her I'd drive it up there, and she said she'd give me a drive in her yellow convertible Mustang.
Oh.
Right, Elena? Is that a yellow Mustang
You
convertible?
should always ask to drive it.
I don't want to drive it,
Why
but
not?
let her drive me around.
All right.
It's much cooler to have a woman drive you around in a car. Didn't you know that?
Do we have an address, and then we'll just price out how we can get.
I
it
do
there.
have an address.
All right.
She gave it to me, so I'll give it to you. John says the only guarantees in life are now death, taxes, and herpes. Yes, it is a guarantee. I think that was another joke that my friend Saul Silverstein used to say. Okay. Vincent, do you wear a mask on planes when you travel? I'll never stop. I always used to get sick when I flew. I used to, actually, and I stopped. So when the plane is, so Vanity will tell you at some point the ventilation system kicks in. I don't know when that is, Vanity. Tell us. Is it when you're taxiing? Is it on wheels up? When is the thing kicking in? And then I feel it's okay to take a mask off. But if there's somebody hacking away next to me, right next to me, like there was, I think on the way to Austin, this guy next to me was hacking away. I put a mask on.
Yeah.
I think you should protect yourself. I don't know what he had. Maybe he had allergies, but yeah.
See, I wouldn't go on a plane while I was coughing without wearing a mask.
Yeah, I think that's not right. I mean, there was a woman next to me on the subway tonight sniffling, but she was wearing a mask.
Mm-hmm.
I mean, you can hear when someone has got a runny nose, they go all the time, right?
Yeah. Or
But she had a mask
she might have been doing cocaine.
She might have had allergies. Boy, you think the worst. Don't you think the worst? Ariozone wants to know how measles wipes out prior acquired immunity. So the virus reproduces in memory B and T cells, right? So these are the cells that retain a memory of what antibodies to make or what peptides to attack, and the virus reproduces in them and kills them. So you don't have any more memory, and you have to redo the vaccination or the or the infection. The effective herpes meds. Are you talking about valacyclovir or valganciclovir? Not favipiravir, no. But the first two, yeah, valacyclovir or valganciclovir can be used for various herpes viruses. And some people still use acyclovir. Over the counter, you can buy acyclovir, which still works. But so acyclovir is not orally bioavailable, whereas valacyclovir is. So if you have to take it orally, it's a prescription drug. You would take Vala, right? Or Valagan is another modification. But acyclovir would be a topical cream that you could put on a cold sore. And so now it's over the counter because you're not going to overdose on it, right?
Elena did correct you and say it's a red convertible.
Sorry. Well, it's still cool, right?
No, it's way cool.
I'm just buying a new messenger bag, and I almost bought a red one. I thought that would be a bit much, right?
Why?
Oh God, that's just.
I
much.
promote too much all the time. Anytime you have an impulse, go with it.
I bought a black one.
All that only seems like too much till you carry it around a few days, and
Well,
then
I still
you forget.
think these glasses are too much, Karen.
Those
glasses I
glasses.
usually wear, which I can't see the computer screen. I think they're too
Well,
much.
eventually you get used to them on your face.
They're blue, red, and white.
You're not used to them. You need to look in the mirror more then, because then it'll just become a part of your face.
We need a mirror here at the incubator, I think, right?
Yeah, we don't have a mirror in here.
We need to. John donated to Microbe TV this week. So there are two links here that you can go to. You can go to microbe.tv/contribute, or you can go to Venmo at Microbe TV. And I know you don't like that, Karen, but maybe we'll stop putting that up there at some point.
I've got no problem with it. Let me just throw out there my issue, just in case anybody cares, is when we go in to do the bookkeeping, when there are $1 contributions that are every month that people are doing, it often takes us more time to do the bookkeeping than that $1 every month is worth. So my only issue is, this is free for anyone who wants to utilize it. All of our podcasts are free. And if you have resources, obviously it needs to keep running, so contributions are great. But the $1 ones are a big logistical bookkeeping annoyance. So Venmo's fine. But
Okay. B says from Algeria, Karen, all respect to you for your care in choosing appropriate words to describe.
Oh, that was about, I did the other thing. So you can say developing countries, but also what they're saying you could say is the majority world.
Okay.
To reference, when you want to make reference of,
I got
that. You could say the majority world, and that lets us know that we are a smaller percentage.
Jim says, shingles of the eye occurs in about 10 to 20% of people who develop zoster, also known as shingles. Yeah. Okay. That's too bad. Where was the other one? So Doreen says, when you say shingles in the eye, are we talking about the eyelid, socket, or eyeball? But Jim's didn't say exactly where it would be and which cells. But, herpes is on the epithelial cells lining the cornea, the outer side of the cornea, right? And someone wanted to know who was talking. Rob wanted to say, who is talking? And that's you. James loved the twin video, so we should do those again, Karen, okay?
Oh, I got down.
Very good. Steph says you're great, Karen. So I
Thank
guess you
you.
guys like having
I'm going to tell
Karen
you,
here.
you will never make everyone happy, Vincent, so you should just work on making yourself happy.
So Alan says Karen is a perfect foil. She's the spokesperson for us lay peeps. Siddhartha says Karen is super. Steph says Karen is great. So you got good reviews here. It's good.
You just wait till you'll get one that'll say.
Elizabeth says it adds a dimension to the chat. Where is that? Here it is. Elizabeth says it'll add a dimension to the chat. James says, Karen, you rock. Emily says, we deserve 1,000 people. Your channel should be number one on YouTube. Isn't that nice?
Yes.
Isn't that nice? No, we don't entertain you enough, I guess. Greenfield says you're splendid. Bernard says he likes having Karen here. Okay, so you're a winner, Karen.
Woohoo,
Karen
chicken
can be
dinner.
your Ed McMahon, according to Craig. Dennis says, Dennis is a TV producer in Nebraska. He says she has a magnificent voice. She could do great voiceovers. Didn't you used to?
Yeah, I did. I did until the industry itself went to all like pay for play, do your home studio thing, because I enjoyed going into and playing with engineers and directors and producers and going into the space. It just doesn't happen that much anymore.
Here's a virus question from Frank. I have been reading about viruses that move from one kingdom to another, plant to fungus and vice versa. Do you know of any other kingdoms that viruses can switch to? Plant to fungus and vice versa. Well, there's some... Depends what you mean by the kingdom. Let's say, what are the kingdoms first?
Isn't that show on how the zombies are going to happen? Isn't that?
So we got fungi, plants, animals, protozoa, bacteria, archaea. So there can be, there are thought to be some changes or shifts of viruses between animals and, insects are animals, so that doesn't really count. But yeah, the plants, fungi, and animals, I think those are the only ones I'm aware of. Now, of course, eukaryotic cells are probably ancient archaea, right? So they brought their viruses with it. The mitochondria is basically bacteria. Those viruses came with us. But we don't see any modern day this sort of things happening. You're my Ed McMahon, apparently.
That I'm going to have to go back and watch some Johnny Carson. It's Johnny Carson, right?
Karen said it's like the night show host talks to the guy in charge of the band. Yeah, that's right.
Yeah.
So 3 people have answered my question. If you were alone with me, what would you do? John says, I would ask for as much instruction through all the facets of microbiology as I could get through for the rest of my life. Thank you, Abdul, for your contribution to science communication. I appreciate it. How do I get on the list to see you guys when you are in the Austin area? Well, we just were there, James. Sorry.
Okay, so James, long-term plan here is what we're trying to do is we are trying to get where everybody's located, build a database of the greater community, which, quite frankly, involves you guys. It involves listeners. It involves patrons and people who donate, so that we know where people are, so that when we go on the road, we can reach out and say, hey, we're going to be in your area. And, pool resources with you, or have you let us know if there's someplace else in the general area that we should be recording a podcast. So that's the big plan. And as a general rule, if you want to be within the context of this database, you can send your address to incubator@microbe.tv.
So Elena from Coxsackie, New York says, I have not wandered off. As a non-scientist who only found viruses during lockdown, the only podcast I haven't given up on, because it's real ass science. That's us.
Nice.
That's us. Let's see. Tom used to teach math models of virus population dynamics. So we could have an epic hour, Vincent. And Richard Schoenbaum, DDS, who often writes, Karen is a great addition to science communication.
Sweet. Thank you. I'm getting all...
So
I'm blushing.
B from Algeria wants to know, explain virology to me and why we can't overcome them. Oh, man, that would take like a lot of time. If you want to learn virology, just watch my virology lectures here on YouTube, okay? And we have done some really good things in virology in terms of overcoming them. We have a bunch of great vaccines that really work. The COVID vaccines are amazing, developed in 1 year. Amazing. We have a lot of antivirals. There's still a lot to go. But I would say we're never going to overcome them. We're never getting rid of them for the most part. They're always going to be with us. The question is, how can we live with them peaceably? And we're learning how to do that. We need research to do that, right? And the U.S. has a great research budget, but it's not enough. The NIH budget is $37 billion a year. It's a drop in the bucket. But I know NASA's only about $18 billion, and they put rockets up there. That's pretty cool. But we all need more money. And science is the big return, folks. It keeps you healthy and living. You got to support it, as well as science communication, of course. Elena says, I wish I could edit my previous comment to only found a fascination either viruses during lockdown. That's fine. You can do that. You can edit it.
We understood what you meant.
So Lise, I asked Lise how she avoids stress or whatever. She said, I work out, meditate, manage my calendar well, have an occasional martini to manage my stress. I was doing fine until Donald Duck's name came up tonight. MK writes, the first, second, third world designation goes back to the Cold War era. U.S. and aligned nations were first, Soviet bloc was second, and non-aligned nations were third, now rather out of date. Good explanation.
Wait.
That's from MK.
I'm trying to find it so I can write it all down.
Tom says, did someone say Janis Joplin?
Yes.
Yes. Karen used to do a Joplin cover, right? Would you say that?
In my old life, I did a cabaret show where I sang 13 Janis Joplin songs, and I told this story about how I discovered my voice. But this crowd is probably more interested in the fact that I used to be a human director of a trained cat circus at one time.
Oh, my gosh. Noir LeBlanc from Santa Fe is going to love that. Well, she was here for TWIV 1000. By the way, you did a couple of Joplin songs in Germany, remember?
Yes, I did.
I don't think anyone recorded them.
Nah.
Very Nika says, in Moscow, where did it go? It always jumps up at the end. One can order Paxlovid for $70. She says a version. I guess it's bootleg. But does it help with death prevention? Nope, even docs don't know about it. Where is it? Even docs don't know about it. Well, regular people don't trust this medication. That's not surprising, right? Not surprising. Oh, so his name was removed. I'm sorry. So my source of comments are the streaming software, which it doesn't respect the comments removed by the moderators from the actual stream that everybody is seeing. So sometimes I pick a comment and I don't realize it's been deleted. And I don't mean to override a moderator. It's just I look at it and feel like I want to talk about it, right? All right. Let's see. Tom says, fanboys. Oh, I pestered Vincent at ASV 2022, and he was generous with time and conversation. And if there was just a few of us here, I'd drop the blue wrench and pester more. In other words, he wouldn't be a moderator, but he'd like to ask more questions. That's cool.
Yeah, it is hard to do both, isn't it?
It is. Scientists are, according to Elizabeth, science are regular people too. We're all just folks. It's true. We're all just folks. All right, let's see. Now, we should start wrapping up because it is after 10. How did a guy in Germany ever manage to get more booster shots? He just apparently gamed the system. He could sign up for a vaccine all throughout Germany, sometimes 2 a day in different places, and they didn't keep records. Apparently not. Yeah.
And anything exciting happening this week that you're looking forward to? You want to let them know before you go?
What's happening this week? I'm just looking at these questions. They're really good, and I hate to pass them up, but I'm going to thank Gabriel for his contribution to support science communication from Mexico. Thank you so much.
Oh, yeah.
We really appreciate it.
And Steve?
Yeah, I'll get to Steve. Yeah, I'm looking at all these other questions. Science comedy trio, Vincent, Dixon, and Allen. I like doing solo comedy, right?
Vincent, Dixon, and Allen.
It's not, we need a woman in there. Yeah.
Though it does have a nice ring to it. Vincent, Dixon, and Allen.
B wants to know if you can if herpes spread from your mouth.
From my mouth?
No, if you have a herpes sore on your lip, is it going to spread to other parts of your face? I think unless you're severely immunocompromised, it will not. But in a severely immunocompromised person, you can get lesions everywhere.
But
yes.
You can spread that to genitals
your
mouth.
You could.
Yeah.
Yes, you could. So Garrett is here. Enjoyed your visit to Austin. I felt bad when Rich showed his spike shirt and I left mine at home.
I got the most beautiful picture of Rich in that spike shirt. He looks like a superhero, and it was a complete accident.
Yeah.
Did we do anything with that yet?
We haven't done anything.
It needs to be something because he looks like a superhero. It was the first frame when I turned the cameras on was him showing off the shirt.
Someone else from Austin is here also. I missed his name.
You can search in that screen.
Jamie Gill.
There you go.
Do you remember Jamie? I think he came back and had dinner with us. Yeah. So Jamie, I don't know where your comment is, but thank you for coming to Austin. Really appreciate it. This is an interesting question. Does measles get rid of autoimmune disease in an individual? I don't know the answer to that. I don't know if enough patients have been studied to know that. The few studies they've done is 70, 100 patients. I don't think there were any immunocompromised people among them. But it's an interesting question, right? And it's a two-edged sword, a silver lining to a black cloud, or all those things. Good question. Steve, thank you for your contribution to science communication. We really appreciate it.
Thank you, Steve.
And so someone asked about why mRNA vaccines aren't adjuvanted. And I think the nanoparticle is an adjuvant in itself. And Ellen says, I don't see how an adjuvant would help an mRNA vaccine. An adjuvant's purpose is to recruit immune cells to the injection site, which isn't where mRNA does its thing, but actually the mRNAs are they're picked up by APCs, right? I mean, macrophages and dendritic cells. And so it's my understanding that inflammation would give you a better adaptive response, right? Communication between innate and adaptive. So I think that the nanoparticle in itself is a kind of an adjuvant. Right? All right. Yeah, a lot of people think you have a great laugh. A yellow convertible muscle car. No, it's red. It's a red convertible, right? Or it's just a red muscle. It's a red Mustang, and damn, so accurate on exit 21. Yes,
right?
Because I took a picture of that driving by once, and I'll never forget. I show it to my classes. Exit 21, New York State Thruway. Some things I remember. Red Mustang, I didn't. Vanity. Here's Vanity's answer. Oh, by the way, let me do this. Vanity says when the plane leaves the gate, the air system is on. Thank you. And she says when people are walking down the aisle, you should wear a mask because all those people one by one walking past you, right?
Ah, yeah.
Right. And Will says a study has shown that 96% of flights have SARS-CoV-2 in wastewater. Yeah. There was a study done in Belgium, and I met the author last year, and I mean to have her on TWIV. Make this note.
Okay.
Get Belgium author of this study where they got planes from China landing in Belgium, and they took the wastewater from the plane, and they looked for virus, and they found SARS-CoV-2 in all of them. And they could do that because once they land in Belgium, it's their crap water. They can do whatever they want with it, and China has no say in the matter. That's very cool. I want to get her on TWIV to talk about that.
Okay.
Yeah, so here is probably good to wear one during boarding, especially with everyone walking past your seat. And Lee says, if measles wipes out prior immunity, should one be revaccinated? Yes. If you've had a bunch of vaccines and infections and you get measles, you should get those again. So it's important to have a good history of what's going on here, right? This week in muscle cars. And Pete says you can turn a monthly sub into an annual one. That'll help with your bookkeeping issues, right?
A monthly
into an annual. So if you give $1 a month, you could do $12 a year and...
yeah, other people can do that. Yeah, we can't change it.
No, they can do it themselves, right?
Yeah.
Okay.
Yeah, we're going to start encouraging...
probably measles only clears immune memory in severe disease. No, actually, in the paper, they looked at kids with severe and moderate measles, and they both had a wipeout. But it's not 100%, right? It's a certain percent of all the T and B cells against epitopes, certain specific virus epitopes, right? And so it's not 100%, but it's enough to make a difference, right? Okay, Karen's volume might be a little high. I could play with that later. Yeah. Okay. Marco wants to know, how should I clean a stainless steel water bowl accidentally left out overnight? It's my dog's. Is bleach enough? If a bird took a bath in it. Yeah, I bleach. Absolutely. That'll kill it. That'll inactivate it. Mm-hmm.
Oh, we got a request to bring you to San Francisco.
I would like to go to San Francisco. I would like to... I'd like to go to San Diego to, interview Stephanie Strathdee. And then we could go to San Francisco at the same time. Here is Jamie, who we met in Johnson City and University of Texas, Jamie Gill.
Wait, did you say San Diego and then go to San Francisco?
Oh, I lowered you too much. Talk.
Did you say go to San Diego and then go to San Francisco?
Yeah.
Because those are like on opposite ends of the country.
So no, they're both opposite ends of California, right?
Right, which is...
Or not quite opposite, but
yeah,
it's not far. Can you go 6 hours out there?
That's like you're saying we'll go to South Africa and then we'll go to Frankfurt afterwards.
Yeah, right. All right. Anything else here? Let's see if we have... Laura says we need to have longer shows. I don't mind having longer shows. It's already 10:10, but once I did a 3-hour show, and people loved it. But I had to warn the moderators because it's not fair to just say you got to stick around for another hour.
Oh.
So we won't do it today. But I love answering your questions. And Alina says there will always be more questions. You got to rest at some point.
Right.
Could we get Alan for office hours? I don't know if he would do it. I've asked a few of the TWIV hosts. I can ask him. Should I get Alan for office hours?
I'm gonna write it down.
And Very Nika says Alan on office hours will be this week in airplanes. Yeah, that would be fun. He and Vanity could talk about airplanes. All right. Let's see. Peak. Hello, Peak. Our other moderator is here at the end. Peak, hello, welcome. You're a busy guy and you don't have time to moderate. That's just great. But welcome. And
Flight time
Happy
from
to
San
have-
Diego to San Francisco is under 2 hours?
Yeah, it's close.
I guess I've only ever driven it.
So Tom says if I'm going to go to San Francisco, then I have to go to Portland, Oregon. I have to make a West Coast
Right,
trip.
exactly. That's what I was thinking. I was thinking
All right, we'll make
San
a West
Francisco
Coast
is
trip.
like Portland, Seattle, as opposed to San Diego, I think of as, Southwest.
Peak says, I had the lucky opportunity to meet one of Professor Racaniello's colleagues, Professor Dasgupta. Gave an awesome lecture on coronaviruses. Yeah, we were postdocs together in the Baltimore lab. Oh, we have a late night edition from the Philippines. Thalia 7, welcome. Steph says it's a 1-hour flight from San Francisco to San Diego. So we can go 1 hour and 40 minutes. There we go. All right, we got requests for Alan on office hours. And Mr. Doreen says, meeting Karen in Chicago was a joy. Do you remember meeting Mr. Doreen?
Was his name Mr. Doreen?
Oh, this is his wife's first name.
Yes, I thought so. I think...
He had a picture, holding up a picture of his wife, remember?
Yes.
You met him. That was fun.
Oh, that was great.
The virus tour. Yes, let's end it with that, the virus tour. We have to do a virus tour.
Yeah, working on it.
All right, I think we'll call it a night here. I want to thank all of our moderators for joining us. Let's see, who do we have? We had Steph SF.
Mm-hmm.
We had Vanity Nutrition.
Mm-hmm.
Pete Dunning-Kruger came at the end. We had Andrew.
Mm-hmm.
We had, at the very beginning, Frank was here. Tom Steinberg was here. And I believe that's it, right? That's all the...
That's all
moderators.
I saw.
Oh, and you're a moderator now too, Karen, but you probably haven't been moderating. Right?
No, it's very hard to multitask like that. They're right. If they can actually even write things down and moderate at the same time, it's because they're super geniuses.
I want to thank everybody for coming. We had almost 200 people here, and appreciate your likes and your support. And let's see, what's the schedule? Is next week another office hours? Maybe Alan can come next week. I'll have to ask him right away. Next week is the 3rd of April. So, and that's the 3rd of April. We do a TWEVO live stream. So mark your calendar. And live stream office hours at 8 p.m. I'll try and get Alan. We'll see what we can do. But also remember this, when you said you want to point out anything that we're doing. So Friday we have Paul Offit on TWIV to talk about his book.
And that'll be released on Sunday morning,
which
at
is
midnight.
March 30th or something like that.
Oh, am I supposed
I
to
think
have a calendar in
I
front
think
of
the
me?
first is April Fool's Day. Here's Paul Offit's book. Tell me when it's over, and he will talk about why he wrote it and what's in it and so forth. That should be a lot of fun. And otherwise, folks, stay safe until next week. But I do expect to see all of you here next week. We will not have a false start to this live stream next week. We always learn from our errors. Thanks for coming.
Thank you all so much. This was such a pleasure.
Thank
what
you, Karen.
a lovely little crowd you've got.
Yeah, we did build a cool crowd. I thank you all for coming and coming every week. It's amazing. I didn't get a complaint about the blue glasses. Karen, thank you for coming this week. Appreciate it.
Oh, my pleasure.
Thanks, everyone. See you next time. Good night.